with the steps of the transcription one by one. Let's start with the first step, which is the initiation step. Initiation is the step in which the RNA polymerase enzyme binds to the DNA. As the DNA is a long sequences, to which region these RNA polymerases will bind. So for binding of the RNA polymerases, there is a specific sequences which is known as promoter. So promoter are the nucleotide bases or sequences to which this RNA polymerase will bind and will initiate the process of transcription. And before initiation, there are some sequences which you can also call them pre-initiation sequences. So promoter, it contains the resequences to which this machinery or the transcription machinery will basically binds, and then this is basically the coding region, then followed by the coding region which will be copied by messenger. As you can see here, this is the template strand which is copied by the messenger RNA, and this is the sense strand. But from where the transcription has to begin, how would this RNA polymerases will know? RNA polymerase will bind to this promoter region, also act as a regulatory region. When the transcription is required, it will bind to this promoter region, and when the transcription is not required, this regulatory region will turn off. So these are the promoter sequence. The promoter sequences, what are the sequences basically which will be recognized by the RNA polymerases for transcription? These sequences are Tata box. Tata box is present -10. These are the sequences. This is the promoter region. Most commonly we say that this is the upstream region, and then this is the downstream region. This is basically the site from where the transcription starts. So this is the initiation start of transcription. This is the initiation step. We call them +1. So from 1 + 1, basically this is the region to which the RNA polymerase will bind, and then 5' untranslated region. This is basically the coding sequences region, and then 3' untranslated region. From this initiation start site, the upstream, there are sequences that varies in different organisms. It may vary from, these sequences may vary from 100 to 1000 sequences. The most important Tata box is in prokaryotes, it is present -10 upstream, while in eukaryotes, the Tata box is present -25 base pair upstream. As you can see here, -25 base pair upstream in eukaryotes, the Tata box sequences, and this is the TA and TA sequence, and then -80 upstream base pairs, when you go upstream 80 base pairs, then -80 upstream base pairs, that's the CAT box. You can see cytosine, adenine, thymine box, and then -100 means that 100 base pair upstream, there is the GC box. And there is also in prokaryotes -35, or you can say 35 upstream base pair sequences. So these are the specific sequences which are recognized by the RNA polymerases, and RNA polymerase binds to the sequence, and from there then it starts the transcription, initiate the transcription, and then it follows the coding region from where these codes are copied and then carried to the translation.